Hello, everyone, and thank you to the organizer for giving me the possibility to speak here today. I will present you napari-morphodynamics. I'm Stojiljkovic, and since 6 months, I am bioimage analyst. So I'm in the field since 2017, actually, when I first did the new bias in Lisbon, and I fell in love with this bioimage analysis community and this job. So it took me only 6 years to be here, going through the entire process, PhD, postdoc, and microscopist at some point. And very recently, I joined the Data Science Lab. So basically, it's a new core facility at University of Bern that provides support and training in all questions about data science. And Guillaume Witz and me are responsible for all the questions about image analysis. So this to say that actually we don't produce our data. And I also want to hear acknowledge that because I joined only 6 months, I haven't done most of what I will present today. So I'm very honored that I can present the work of our lab. And the data, we actually rely on collaboration with scientists. And one of our main collaboration is Olivier Pert's lab. And what they do actually is, since many years, they study morphology and dynamics in single-cell time-lapse movies. So this is a typical image that you can see is a DSC on the right. And on the left side, we see this dynamic range. So basically, we can have different biosensor, and one means, well, this biosensor doesn't move or vary a lot during that time-lapse, and we see that in the protrusions, we have some variation. This means it's very local, what is going on here, the biology. And since shortly, they also start doing a lot of optogenetics. So what you can see here is this movie that I really like watching is hypnotic. So they basically stimulate the cell with optogenetic tools, and then they create waves within the cells, or they can kind of control very precisely the movement. So they shine light, and then the cell will follow this light. So this is the type of movie that we work on. And this means napari-morphodynamics is very specific to single cell analysis. And the title of my abstract was napari-morphodynamics, quantification of cellular dynamics made easy. So when I wrote it, I realized that this means that it's not easy. So there are some challenges. And the first one, I think, is segmentation, as we have seen in many talks. So here, particularly, we want to segment very round cells that then become very elongated, but we also want to be very precise in the protrusion, because this is where the biology happens. Then we want to kind of segment a smaller region of interest for subcellular analysis, and then follow these windows around the cell. And the data analysis become complicated, because now we want to kind of correlate movement with some biosensors that we can monitor in a cell. And then we have seen yesterday with QPAT actually what we have discussed. If your tool is not used, then it's maybe not useful. So we want to wrap it up also in something that is very accessible to biologists. And the last challenge, I think, is scalability, because, of course, I show you single cells, but we do it over time lapses, so it's 300 or more frames, and we want to do it like for many cells, because it's then that we can quantify biology. So how we tackle the first problem of segmentation? Well, we have many different tools, and these are only examples. So what we allow in napari-morphodynamics is basically to import any type of segmentation that you have done previously. We also have in napari-morphodynamics access to the cell pose plugin, so you can perform cell pose analysis directly in morphodynamics, because we don't really want to reinvent the wheel. And do we? Well, maybe a little bit. So we want to make it a little better, maybe. And this was the idea of Lucien Hinderling, a PhD student in Olivier Pert's lab, and we really love Elastic. And this is a random forest classifier. We use it a lot for pixel classification, but for those who have used it, I imagine many of you, The first step when you want to do pixel classification is select these features. And when I was very new in the field, they will tell me, well, you check some boxes, you try with everything. Well, if then it's computationally heavy, maybe you reduce them. So how do I select how many features I really want to measure and which one are the really important one? So Lucien came up with the idea, why don't we use a convolutional neural network like VGG16, and we use some of the extractor feature from this model. So this model does feature extraction and then classification. So for example, you show a picture of a cat and it says, is a cat or dog, guinea pig, and it's trained on millions of images from the ImageNet dataset. So the idea is, why don't we drop the classification part and we use the feature that are extracted anyway from this model to then put into a random forest classification. So we annotate few pixels and it should perform amazingly. So this was the idea. And he got funded by the Chan Zuckerberg initiative. And here is the architecture. So we input images, but we also input it at different scales because some features are easy to recognize in smaller scales. So we downscale the image, we put it in the model, and then we extract these features. And then we upscale everything to the same shape so that we can feed the random forest classifier. And the idea is actually what is great in what they have done here is actually they built a backbone. So now instead of VGG16, you could use any model. And yeah, for example, we were thinking of Dino V2 because this can recognize the head and the tail of a dog. So maybe it will be able to recognize the moving edge, so the protrusion and the tail following an inside. So this is what we are working right now and testing it out. And this is only like a small demo. Here is not biology, but the floor of the main building where the plugin was built. And here you see with very sparse annotation, I can quite quickly classify 5 different classes. So it's not only for biological images, it's for any image that you can think of, and it's quite fast. So this is napari-conv paint, so convolutional and paint, to remember this, and you can download it and try it out. So it's a plugin per se, but it's also the default segmentation that we use in napari-morphodynamics. From the point of view of the windowing and data analysis, this we didn't invent something new. This was already done many years ago by a collaboration between Oliver Pertz and Gaudens Danuser. So basically they have developed this windowing to study protrusion. You segment the cells in small region of interest and then you follow them. And then now you can do many type of correlation. You can look, for example, at the velocity and then compare how the biosensor correlate with this velocity. So you can see which proteins are important in this dynamic, so morphological changes of the cells. So these were already all built, and it was in MATLAB. So to make it more interoperable, Guillaume Witz, who is, I would say, our orchestrator, so he kind of is coordinating all this work that we are doing. He did a lot of work to transform this in Python because we are now in a quite Python-based world and we wanted to make it interoperable. So he built a lot of Jupyter notebooks. And you see that the analysis is quite complex. So he will start sharing these long Jupyter notebooks. Then he divided it in many Jupyter notebooks. And then, yeah, biologists got very scared and didn't use it. Really. So he got funded for wrapping up in a nice Napari plugin because Napari became quite popular and very flexible and useful. So he did it. He wrapped everything together in Napari, and he also made it scalable because he implementing DAS computing to be even faster, and now you can also deploy it on a cluster. So here is a short demo if I have still time. So we see the cell, 2 channels, and what we do here, we use conv paint to segment the cells with few brush strokes, and you see that the protrusions are segmented quite well. And then we apply this windowing. So you will now see these windows appear, and we can track these windows as well. And we have done it in a kind of user-friendly way. So it's tabular. Every tab has like a job. You do data import and segmentation. Then you do the windowing. And then, for example, you can display all these plots that I've shown you within the plugin. So this is napari-morphodynamics. And then here is what I've done. So if you remember this cell that I showed you, well, it will be a little bit computer overkill to try to window this and track it because the cell is not really moving. So what you could maybe want to do instead of this having very computational heavy software, you might just want to build a region of interest and follow what happens. So this is my first Napari work. And so you have these two channels, and now you can, for example, have a segmentation that I did with ConfPaint or not. So maybe you can add a point of interest. I add it outside the cell just to show how it works. And then you can decide to add some fixed shape. And now I decided to apply it on the center of mass of the mask. So it will move to the center. And now I can do some intensity measurement. And we have here different shapes, as you can see from the logo from napari-morphodynamics. It doesn't have to be circles. It can be, pies. And so we can implement more shapes if you wish. Just try the plugin and let us know what feature you want more. So thank you to the Data Science Lab. Here I've put the code of Guillaume Witt because you can find all of these plugins on his account, but also because he's a very good teacher. So if I can stand here today talking about Python, because he is very good in explaining Pythons. He has a lot of material for training. This is really, I'm advertising it because I like his job, not because he's my boss. And I want to thank the Napari community because it's amazing, the documentation. Again, I wouldn't have been able to do this. And here at CRIT, I want to thank Rocco D'Antuono. We also organized a few months ago, like a very spontaneous hackathon comparing plugins. So it was a lot of fun. So, and thank you for your attention.
I think we have time for one question. If not, then yeah, you can find Anna afterwards.